Welcome to the deep dive. Today we're tackling something really fundamental. How our brains learn to let go of fear. Think about those moments when a threat passes that feeling of relief. It isn't just like anxiety fading.
Right.
It's your brain actively signaling all clear. We're diving into some recent MIT research that's trying to pinpoint exactly how this works.
Yeah, and focusing on a surprising player, dopamine.
Dopamine, exactly.
That's right. The sources we're looking at, mainly this fascinating study done in mice, they reveal a very specific dopamine-driven reward signal. And this signal seems to be right at the heart of what scientists call fear extinction, basically unlearning fear.
Okay.
So our goal today in this deep dive is really to unpack this mechanism and, explore what it might mean for understanding, maybe even treating things like anxiety and PTSD.
And the really interesting angle is this idea that overcoming fear isn't just about shutting down a bad memory.
Right. Precisely. It seems to be an active kind of positive learning, and it's fueled by a reward signal, which is quite unexpected in this context.
Okay, so let's get into it. The core question is, after something scary happens, what actually flips the switch in our brain to say, okay, you're safe now? And this new MIT work is pointing strongly at dopamine, playing a key role in that all-clear message.
Exactly. The researchers, they really zoomed in on a specific circuit involving dopamine and the amygdala.
Ah, the amygdala. Most people have heard of that, the emotion center, especially for fear.
Right. And their findings suggest that when this dopamine pathway is, working correctly, it actively promotes a sense of safety, of calm, after the threat is gone.
And the flip side, if it's not working right.
Well, conversely, yeah, if that pathway is malfunctioning, it could be a big reason why anxiety lingers or even why something like PTSD develops.
Dopamine. We usually associate that with pleasure, reward, motivation. So the idea that it's also involved in calming fear feels a little, unexpected. Can you explain that dual role? It sounds almost contradictory.
Well, that duality is actually what's so insightful here. The research shows dopamine isn't just released when we, say, sense danger or anticipate a reward in the usual sense. It also gets released to reinforce what they call positive learning.
Positive learning.
Essentially learning that a situation, maybe one that was threatening before, is now safe. And it seems to do this by acting on different groups of nerve cells in the brain quite specifically.
Okay, which I think brings us to some earlier work from the same lab. There was a 2020 study you mentioned.
Yes, that's right.
That identified these kind of competing groups of neurons within the amygdala itself. How does that older research connect here?
Yeah, that 2020 study was really crucial groundwork. It pinpointed two key populations of neurons in the amygdala that are, you could say, competing when it comes to fear. So the first group, they're in the anterior part of the basolateral amygdala, the ABLA. Anterior is just meaning the front part.
Okay.
And these neurons express a gene called RSPO2. They are critical for storing fear memories.
Storing the fear, like the initial shock.
Exactly. Imagine a mouse gets a mild foot shock in a specific box. These RSPO2 neurons help link that place with danger. They create the fear memory.
Got it. So the RSPO2 neurons are like the brain's little danger here flags. What about the other group?
So the other group is in the posterior part, the PBLA, posterior meaning back. And these express a different gene, PTP1R1B.
PPP1R1B.
Okay.
And these neurons are involved in encoding the extinction memories, the safety learning.
Ah, so they're the ones saying, actually, it's okay now.
Precisely. If that same mouse goes back into the box and nothing bad happens, these PPP1R1B neurons start to kick in. They begin to override that original fear memory, teaching the brain, this place is safe now.
So it's a competition.
It really is. That 2020 study highlighted this sort of tug of war between these two neuron groups as being central to how we learn to overcome fear. Right. And this new study, then it takes it a step further. It asks, okay, what actually triggers these different groups? What tells the RSPO2 neurons store fear and the PPP1R1B neurons store safety? What's the starting signal?
Not exactly the question. The researchers wanted to figure out the initial prompt. What makes them encode these different memories? And their big discovery is that it's dopamine. But crucially, it's dopamine delivered to these two different amygdala populations from distinct groups of neurons back in the ventral tegmental area, the VTA.
The VTA, right? That's a major hub in the brain's reward system, often called the pleasure center, though that's a bit simplistic.
Very key for reward processing.
So how specific are these connections? Is it like a dedicated dopamine delivery service to the fear neurons versus the extinction neurons?
It seems to be incredibly specific. They used some very precise circuit tracing methods, and they found that the ARSPO2 neurons, remember the fear memory ones, they get dopamine mainly from neurons in the front, the anterior part, and also the sides, left and right of the VTA.
Okay, front and sides for fear.
But the PPP1R1B neurons, the fear extinction, the safety ones, they're targeted by dopamine neurons located more in the center and the rear, the posterior parts of the VTA.
Center and rear for safety. Interesting.
And another really interesting observation, they found that the PPP1R1B neurons, the safety ones, actually had a higher density of these dopamine connections, more inputs.
So more dopamine highways leading to the safety learning neurons.
It looks that way. It's like the safety learners have maybe a more direct or more potent line to the dopamine supply.
So it's definitely not just a general dopamine flood in the amygdala. It's highly targeted.
That seems to be a crucial point, yes. Highly specific targeting.
And did they look at the receptors? I mean, do these different neurons respond differently to the dopamine they receive?
They did. They found that both types of neurons, the ARSPO2, fear, and the PPP1R1B, safety, express dopamine receptors, specifically a type called the D1 receptor.
Both have D1 receptors.
Yes, but, and this fits nicely with the connection density finding, the PPP1R1B cells, the safety ones, had significantly more of these D1 receptors. So they have more connections and more receptors.
Exactly, which suggests they might be more sensitive to dopamine, perhaps more easily activated by that safety signal when it arrives.
Okay, this is painting a much clearer picture of the wiring, the receptors. But how did they actually see this happening? How did they watch dopamine influencing these neurons in, real time?
Right, because mapping is one thing, seeing the dynamics is another. They designed this really neat 3-day experiment with mice, and they could actually monitor dopamine activity in the amygdala while the mice were learning.
3 days, what happened?
So day 1, the mice got mild foot shocks in a specific box, classic fear conditioning. And when they measured dopamine during this...
Let me guess, more activity near the fear neurons.
You got it. A larger dopamine response was seen near the ARSPO2 neuron population, the fear memory guys.
Okay. Day 2.
Day 2. Same box, but no shocks.
Yeah.
So initially the mice show fear. They freeze up, right?
But over about, say, 15 minutes or so, they start to relax. They move around more. That's fear extinction happening. They're learning it's safe now.
And the dopamine signal during that.
During that period, they detected stronger dopamine activity, specifically near the PP1R1B neurons, the safety learners. And what's more, they found a direct correlation. The mice who showed the strongest fear extinction, the ones who learned safe the quickest, also had the greatest dopamine signal in those PPP1R1B neurons.
Wow. Okay, that's a strong link.
Yeah.
The more dopamine hits the safety neurons, the better the fear unlearning.
It's a very compelling correlation, yeah. It really points to dopamine driving that safety learning.
But correlation isn't causation, as they say. How did they nail down that dopamine was actually causing these changes in fear learning, not just, happening alongside them? That's the crucial step, isn't it?
Absolutely. Establishing that causal link is key, so they use some pretty advanced tools, including optogenetics.
Right, using light to control specific neurons.
Exactly. They could use light to either activate or silence specific sets of neurons with incredible precision.
So what happened when they messed with these dopamine pathways?
Well, when they quieted those dopamine-releasing neurons in the VTA that specifically connect to the PBLA, the safety neurons in the amygdala, they found the mice really struggled to extinguish their fear. They stayed scared for longer.
Makes sense. Turn down the safety signal, fear sticks around. What about activating them?
Conversely, when they artificially activated those same dopamine inputs, the PBLA, fear extinction happened much faster. The mice learned safe more quickly.
Okay, boosting the safety signal speeds up fear unlearning. Direct control.
But here's a really surprising twist. They also tried activating the other pathway, the VTA dopamine inputs going to the ABLA, the fear memory neurons.
What happened then?
Activating that pathway actually brought the fear response roaring back, even when there were no new shocks. It reinstated the fear.
Whoa. So activating the dopamine pathway to the fear neurons reinforces fear, while activating the one to the safety neurons promotes safety.
That seems to be exactly what's happening. It underlines this very specific dual role of dopamine depending on where in the amygdala it's acting.
That's quite something. Did they also manipulate the receptors directly?
They did. They played around with the D1 dopamine receptors on the amygdala neurons themselves.
And
in the PPP1R1B neurons, the safety ones, If they overexpress the receptors, basically making them extra sensitive to dopamine, it actually impaired the recall of the original fear memory and boosted extinction.
So more sensitive to safety signals makes it harder to remember the fear.
Seems like it. But if they knocked down or reduced the number of those receptors in the safety neurons, fear extinction was impaired. The mice had trouble learning it was safe.
Okay. And what about the fear neurons, the ARSPO2 cells?
In the ARSPO2 cells, when they knocked down the dopamine receptors there, the mice actually showed less freezing behavior, less fear response.
So less dopamine sensitivity in the fear neurons reduces the fear expression.
Exactly. All these manipulation experiments, optogenetics, receptor changes, they all converge to strongly support a direct causal role for these specific dopamine pathways in both encoding fear and driving its extinction.
Okay, so stepping back, the really big takeaway here seems to be that unlearning fear isn't just a passive fading process. It's not about suppressing a bad memory.
No, it's much more active than that.
It's an active process of positive learning, right? Where the brain's reward system, using dopamine, is effectively signaling safety and helping to rewrite, or at least override, those old fear associations.
That is absolutely the core insight. It really reframes how we think about overcoming fear. It's not just about trying to forget or ignore something scary. It's about the brain actively learning something new and positive safety and being neurochemically rewarded for doing so.
And the implications for, say, anxiety disorders, PTSD, they seem pretty significant, don't they? If we understand this safety signal circuit.
Absolutely. I mean, the researchers are rightly cautious, pointing out that fear extinction involves many brain areas. It's complex.
Sure, it's never just one circuit.
But this specific dopamine circuit involving the VTA and these distinct amygdala populations, particularly the PBLA safety neurons, it really seems to be a critical node, a key player.
So understanding it better could open new doors for treatment.
That's the hope. By getting this clearer picture of how dopamine modulates these specific neurons, it might lead to more targeted treatments for anxiety, for PTSD, instead of just broadly dampening fear.
Which can have side effects.
Right. Maybe we could develop therapies that specifically enhance this dopamine-driven safety learning process, maybe strengthening that TBLA pathway somehow.
That's a really interesting prospect. So the big aha moment for me, really, is this idea that the brain uses a reward signal, dopamine, to actively teach itself that it's safe.
Mm-hmm.
It's not just fear going away. It's
Yeah.
safety being actively learned and reinforced using the same chemical we associate with pleasure and motivation.
Precisely. It's a really powerful shift in perspective. It moves us beyond just thinking about fear suppression and highlights this active, almost rewarding aspect of building resilience and recognizing safety.
Yeah, it really does. And it makes you wonder, doesn't it, how this fundamental dopamine safety signal might relate to other kinds of learning, or how we regulate our emotions generally.
Definitely.
What sorts of internal cues or even external experiences act as those rewards that help us personally overcome challenges or anxieties in daily life. It's definitely something fascinating to think about.
It really is. A lot to chew on there.
Well, this has been incredibly insightful. Thanks so much for taking this deep dive with us today.